Funding for this program is provided by Annenberg CPD to advance excellent teaching.
There's no denying that chimps and humans are a lot alike. It's clear that we're related. But there's another way to look at it. It's not the shared traits that are remarkable. Rather, given our genetic similarity, how did we get to be so different? Exploring this question through research at the molecular level offers new perspectives on who we are and how the species Homo sapiens may have evolved. Over the years, researchers have struggled to piece together the most accurate evolutionary history possible for the species Homo sapiens. But lack of evidence in the human fossil record has made the job exceedingly difficult. But now scientists employ a range of techniques, even molecular analysis, to propose hypotheses about human evolution and relationships. Traditional approaches combine with newer ones, allowing researchers to fill in some blanks on the hominid family tree. All of this gives us new perspectives on relatedness and a better picture of evolutionary relationships. At the American Museum of Natural History in New York, Ian Tattersall scours the fossil record for clues about the path of human evolution. He has traveled the world to inspect every species in the hominid fossil record. As a result, he presents the true shape of the hominid family tree as more like a bush than a slender, ladder-like progression toward modern Homo sapiens.
Beginning in about the 1950s, the received wisdom in paleoanthropology was that human evolution had been a sort of a single-minded slog from primitiveness to perfection, if you wish, without a lot of different species being involved, and with species tending to evolve from one into another in a kind of a linear fashion. I think the cumulative effect of the new findings of the past couple of decades have reinforced the notion that human evolution is a matter of evolutionary experimentation rather than of a fine-tuning within a lineage. The human family tree, as it's increasingly emerging, is very bushy in the sense that there are many branches, many little twigs, many side branches, rather than a single slender trunk that's sort of proceeding in a majestic way towards the apex. New species are continually produced, are thrown out into the ecological arena, and are triaged, as it were, by competition among themselves and among other elements in the environments they inhabit. And this process involves a lot of extinction as well as of species origination.
But relationships among the hominid branches of the tree are complicated by the diversification that occurred through the process of adaptive radiation.
Adaptive radiation is the notion that when a species enters a new ecological zone, for example, it has the opportunity to exploit this new zone in many different ways. And it's very true that once a new group of animals has become established, it's very common that many species emerge within that group within a relatively short time, all of them exploring the different ways there are to be a species belonging to that group.
Piecing together the hominid fossil record and establishing likely relationships is tricky business. For example, how do you explain seemingly significant features found on one specimen but absent from a likely descendant? Molecular biology offers new ways to approach such questions. At the University of California in San Diego, Christopher Wills uses molecular strategies to explore the emergence of different physical characteristics among hominids as seen in the fossil record. the hypothesis of mosaic evolution attempts to explain the role of genetic factors.
Mosaic evolution is the idea that different genes in organisms, different parts of organisms, may evolve at different rates. Okay, so you've got a situation in which some feature of an organism might evolve very quickly, other features of the organism might evolve much more slowly. So if you look at the hominid fossil record, for example, you'll find that many skulls might have very human-like features and very ape-like features, a funny mix of the two. And it's that kind of thing which has been found most recently in that fossil that they turned up in Chad, which has a rather human-looking face, but the back of the skull looks very much like a chimpanzee. That kind of difference, essentially, between different parts of this animal suggests that perhaps these different features were evolving at different rates, or perhaps were evolving and then disappearing again. There are so many different ways in which you can get a mixture of different types here.
Further complicating the picture of human evolution from both the fossil and molecular perspectives is the fact that Homo sapiens is the only hominid species still alive today. None of our relatives from the hominid family bush are around for comparison.
Evolutionary dead ends are very common in the human fossil record. In fact, there are people who would claim that we probably don't know any direct ancestors to Homo sapiens in that record.
Our hominid relatives have all gone extinct. The Australopithecines were a hominid relative. Homo erectus, the Neanderthals, some of the other close relatives of ours that lived in Africa and in parts of the old world that are now all extinct were all parts of the hominids. So we are the only living member of that particular group.
Many researchers expect that we may never fill in a complete human fossil record. Some questions about hominid species, evolutionary dead ends, and inconsistent physical features are likely to remain unanswered. But scientists are answering other questions about the species Homo sapiens by studying our closest living relative, the chimpanzee. Many recent discoveries were made possible by the genomic revolution, and close scrutiny of tiny molecular distinctions between humans and chimps may help identify exactly how genetic expression contributed to human origins. Molecular differences are the focus of Ajit Varki at the University of California at San Diego. They help him compare genetic expression in humans and chimps, two similar but distinct animals, both living today and both having split from an unknown ancestor several million years ago.
And so it's also important to put aside the sort of common notion that we evolved from a chimpanzee. That's not the case. We shared a common ancestor with the chimpanzee. Chimpanzee has been evolving itself during the last 5 to 6 million years. And what exactly is the nature of that common ancestor is something that people try to predict, but you can't really say what the common ancestor was like.
So Varki worked with an international team of researchers to analyze the brain tissue of humans and chimps. What could the molecules tell us about differences between two species that are genetically so similar?
So this was a study which originated to try and test the regulatory hypothesis. Made in the 1970s, the early evidence available at the time showed that there was hardly any differences that they could find between chimpanzees and humans at the level of gene or protein sequence. And so they suggested the possibility that in fact there may be no gene or protein differences, and that all the differences are due to differences in how genes are expressed. So maybe an analogy one can use is, it's like if you say the genome is a recipe book. The recipe book doesn't do you any good till you decide which recipes to use. So in the same manner, you have to express genes, and not all genes are expressed in all places. So what was done then was to take tissues like liver or blood, where we don't seem to find a lot of functional differences between humans and chimpanzees. On the other hand, the brain is a place where, at least functionally and in size, we'd say there are differences.
To make sense of the similarities and the differences, the researchers also examined an outgroup, the rhesus monkey. which shared a common ancestor with the common ancestor of the chimp and human about 25 million years ago. While chimps and humans have nearly identical DNA, researchers posited that the species differ in the expression of their genes.
We basically obtained chimpanzee and human messenger RNAs from different tissues and qualified them. So it turned out that if you looked at the gene expression differences between the rhesus monkey and the human and the chimp, if you were looking at the liver or the blood, you got the expected pattern. That is, humans and chimps look very similar to each other, and the rhesus monkey looked very different. The rhesus monkey seemed to be the outgroup. But when you looked at gene expressors in the brain, the chimp and the rhesus looked relatively similar, of course with some differences, and humans looked somewhat different. So that favors the regulatory hypothesis that gene expression differences in the brain have occurred, and that these may be partly responsible for the differences in the function of the brain between humans and chimps and rhesus monkeys.
Ajit Varki continues to explore the significance of gene expression and other factors that may have contributed to the evolution of our species. But as humans evolved, they also spread across the planet, inhabiting all the corners of the world. Fossil evidence and molecular data combine to tell the story of how humans came to populate the globe, and how the genetic similarities between individuals today can tell us who we are and aren't related to.
If you pick two random humans from anywhere on the planet, you'll find that they share approximately 99.97% of their DNA. They really differ by only a tiny fraction of 1% of their DNA. And most of those differences, as I say, are noise, okay?
Researchers consider this genetic relationship in the context of the fossil record and draw conclusions about how human species populated our planet.
We're talking here about two different hypotheses about the origin of humans.
Most scientists agree that some early hominid species moved out of Africa and spread across the old world.
At 2 million years ago or so, you begin to find the first hominids of modern body structure, and almost immediately they're out of Africa and they've moved possibly all the way to Eastern Asia. We have evidence of this now in terms of a wonderful fossil record coming out of the 1.7 million year old site of Dmanisi in Georgia in the Caucasus. We have indications from as far away as Java and China at about 1.8 million years ago that humans had moved that far.
But what happened after that is a matter of some debate.
The multi-regional hypothesis suggests that human evolution occurred over a fairly broad front, and that there was a lot of genetic exchange between different human groups in Africa, outside of Africa, between Africa and the rest of the old world, exchanging genes between different human groups so that they all essentially evolved more or less in synchrony.
The replacement hypothesis, also known as the out-of-Africa hypothesis, suggests that modern humans evolved only in Africa, then spread to replace all other hominid species.
The replacement hypothesis, as you know, is the one which is currently favored.
At present, it looks most likely from what we know in the fossil and archaeological records that the point of origin of our species, Homo sapiens, lay in Africa.
Developments in molecular biology lend further support to the replacement hypothesis, while uncovering new information about relationships between human species. One of the most significant and far-reaching innovations has been the identification of a molecular thread that runs through all humans alive today: mitochondrial DNA.
The mitochondria are descendants of free-living bacteria that came to live inside our cells probably about 2.5 billion years ago. The mitochondrial genes are all locked together on the mitochondrial chromosome, and they're passed down from one generation to the next in most mammals most of the time. And that inheritance is maternal inheritance, the reason being that the egg has mitochondria in it. The sperm have mitochondria too, but the mitochondria get lost when the sperm fertilizes the egg.
So, even though the zygote carries nuclear DNA from both parents, it takes mitochondrial DNA only from the mother's side.
And essentially then, as these mitochondrial chromosomes are passed down from one generation to the next, they begin to diverge, not because they're mixing up their genes, but because of accumulating mutations. And as time goes on, these different chromosomes begin to diverge from each other. These different copies of that chromosome begin to diverge from each other. The longer the period of time, the greater the divergence.
Using mitochondrial DNA, which has a faster rate of change than nuclear DNA, researchers can assign a likely date since the time of our divergence from the common ancestor we shared with chimpanzees.
Any human group, essentially, can trace its mitochondrial chromosomes back to an ancestor. Now, that mitochondrial ancestor doesn't mean that we can trace these mitochondrial chromosomes back to some single individual who was the only human being who lived back that time. The mitochondrial Eve is that mitochondrial ancestor. The mitochondrial Eve is the woman who lived perhaps 200,000 or 300,000 years ago, from whom all our mitochondrial chromosomes are descended. But she didn't link up with some Adam and live in the Garden of Eden. She was one of a group of people, and probably a lot of different groups of people living at that time.
Because mitochondrial DNA is inherited maternally, lines with no female offspring abruptly end. Mitochondrial DNA is passed through the mother only, so a male child is a dead end, as is a female child who bears no female children. One line of mitochondrial DNA carries forward to the present from the so-called mitochondrial Eve, a female from a population that lived in Africa only a few 100,000 years ago.
All the other people living at that time, their mitochondrial chromosomes got lost by chance. Hers was the only one that survived. But there might have been hundreds, thousands, tens of thousands of people living at that time, all of whom, by chance, did not pass their mitochondrial chromosomes down to the present.
The chance loss of other mitochondrial lines when human populations were small places the mitochondrial Eve in recent history and helps researchers establish a close relationship among all humans alive today. Additionally, mitochondrial DNA can also single some groups out, helping to verify what many have long suspected. Neanderthals were not our ancestors.
Molecular techniques have added a great deal to the armamentarium of approaches that paleoanthropologists can now bring to bear on the history of our species, Homo sapiens. One way, of course, is by the direct extraction of DNA from ancient humans, which has so far only been done with Neanderthals.
In the 1990s, researchers extracted mitochondrial DNA from Neanderthal fossils to find that it falls outside the range of variation of modern human sequences. Additionally, the Neanderthal showed no closer relationship to European DNA than to that of any other modern human group.
And what that particular work has done has been to draw our attention to the fact that the Neanderthals are indeed very distinctive from ourselves, and it's given us some way of approaching the date of divergence between Neanderthals, or the lineage leading to Neanderthals, and the lineage ultimately leading, on the other hand, to Homo sapiens.
The time of that divergence is estimated at 500,000 years ago, a number that lends credence to the replacement model of human dispersal across the planet. Neanderthals eventually became extinct, giving rise to no further descendants.
The extinction of the Neanderthals is a perennially fascinating question for all human beings, because it's the best example we have of a resident group of distinctive hominids, a separate species, not Homo sapiens, being invaded by an incoming population of Homo sapiens and disappearing as a result. This maybe gives rise to some sort of feelings of sort of guilt. Did we make the Neanderthals extinct? Are we really that nasty that we could do that? Whereas in fact, extinction and replacement of populations and faunas is a very routine thing in evolution. The extinction of Neanderthals is not something we should be particularly surprised about, and it doesn't tell us anything about our darker side than looking around at ourselves in recorded history.
The study of genetic variation among modern humans helps establish our shared molecular makeup, as well as the path of human evolution. But what is the significance of this variation in our daily lives? The people of Iceland believe genetic relationships are very significant. Because they have kept meticulous track of family genealogy over centuries, we can look at the process of human genetic change for clues about genetic variations that may contribute to disease.
Iceland was settled about 1100 years ago. It was settled between the years 870 and 930 by people coming, men mostly from the west of Norway, women coming mostly from the British Isles. Since the country was settled, Icelanders have, for whatever reason, been very interested in the genealogy. We have the genealogy of the entire nation going 1100 years back in time on a computer database. And why is that important? It's important because the study of human genetics is the study not only of the information that goes into the making of man, but also of how this information is passed from one generation to the next. And the genealogy gives you the avenues by which this information is passed. And what we are basically doing at Decode is to take this old story that is basically written in the genealogy of the entire nation and add different chapters to it. The chapter about sort of certain vulnerabilities that are passed on from one generation to the next within these big families.
By matching up genealogical information with DNA collected from the citizens of Iceland, Decode looks for inherited genetic differences across the generations. Volunteers contribute their DNA to Decode's database. Variations found in the sequence are mapped alongside the family genealogy and accompanying medical records to develop pedigrees for inherited diseases found within the population.
SNP is a single nucleotide polymorphism. A SNP is a place where one nucleotide substitutes for another nucleotide in certain proportion of the population. When you are trying to map a gene, one of the things you do is that you take a large number of regions where there is variation in the sequence. And then you look at these variable regions, for example, the SNPs, and you try to you look at a large number of them, and you try to find a SNP where you have more than a chance association between one form of the SNP and the tendency to develop the disease. If you find such a correlation that is statistically significant, it is likely that the gene that you're looking for is in the neighborhood. And by comparing these SNPs or these genetic markers and the various chromosomes in the various genes, you can draw basically a pedigree of human population. Which one came first, which ones are most closely related to each other, etc.
By relying upon the unique circumstance of the Icelandic population, Decode races toward discovery of molecular targets for inherited disease. The ultimate benefits may extend to humans the world over. Decode researchers have isolated genes linked to a number of human illnesses by using a genome-wide linkage analysis of patients. In many cases, such as stroke and rheumatoid arthritis, they have identified the first major genetic factors.
The scarce resource is the population, because what you're studying is the information that is carried in the people. What you need is a population that you understand, that is willing to work with you, and that has a significant amount of information about the relationship pattern that dictates how information has been passed. This is the scarce resource, and we were so fortunate, first of all, to realize this, and secondly, to be able to find the population just by opening, the door to our house that had all of these qualities. What is particularly useful, of course, is to have the sequence of the human genome, because although the sequence of the human genome is only the sequence of the reference person, it gives you sort of the outer boundaries of what you need to look at. It diminishes the necessity of having to do a lot of sequencing yourself. It is enormously enabling.
While the research underway at Decode is promising, it's important to remember that technology can outpace society's checks and balances. Some have accused Stephenson and his for-profit company of pushing the limits of bioethics. Critics caution that some patients may find themselves vulnerable to discrimination or violations of privacy.
I think that this information comes as close to being sacred as any information, so you shouldn't treat it in a frivolous manner. You should treat it with respect, and you should do your best to make sure that it's only used for good. If you ask the question, when I go to a hospital, what is it that I'm using, what resources am I using when I visit the healthcare system? What you're first and foremost doing is that you're using experience, you're using knowledge that was discovered because the people who came before you to this very same healthcare system allowed information about themselves to be used to discover new knowledge to advance medicine. And I think that the case could be made that it is your obligation to allow the information about you to be used, so your children and their children will have access to equally good healthcare system in the future. One thing I think it is, however, important to recognize when we are talking about human disease. If you take all of these disease genes and if you said to yourself, I'm going to develop some kind of a magic gun that allows me to blast away all of these bad forms of these genes, you would probably get rid of man. Because I think that most of these common diseases have to be looked upon as expression of the limitation and design of man.
Studying the design of Homo sapiens won't eliminate all disease, but it may tell us how some diseases work and how we might try to fight them. Such discovery relies upon the continued study of how humans have evolved and how we are related to one another and to other species. And while it's impossible to predict how our 30,000 genes will evolve in the future, we know that change will continue and that the study of genetic change can deepen our understanding of who we are and how we function.
Funding for this program is provided by Annenberg CPB to advance excellent teaching. For information about this and other Annenberg CPB programs, call 1-800-LEARNER and visit us at www.learner.org.